Hi everybody, welcome back. So we're going to start activity 2 now, which is mainly going to focus on the domain bacteria. Okay, so here we've got our domain bacteria. You can see we have all of these different subgroups, some of which you'll see in the supplementary material online under the lineages of bacteria, and some you won't. So bacteria are extremely diverse. We've talked about that a little bit in the last video. Just to reiterate, they're very diverse, very important. Again, they're prokaryotic, they're single-celled, but again, they can also form those biofilms we talked about. Other important characteristics is that they have a fatty acid tail, which is in their lipid bilayer, which is linked to a glycerol by ester linkages. Okay, more about that in a little bit. They also have cell walls which contain peptidoglycan. That's going to be an important feature we'll talk about. And also they have a wide range of life strategies and cell shapes. If you think about the inner structure of bacterial cells, they don't have organelles. They've got a nucleoid region as well as ribosomes, but not too much going on in their inner structures. On the outside, remember they've got the cell membrane covered by cell walls. Some cell walls can even be covered by a secondary outer membrane, which is going to be made of polysaccharides. They can have fimbriae, which are kind of these things sticking out here in this drawing. We can't really see them there. And again, they can secrete polysaccharides for production of biofilm, and they also have these hair-like projections, which can facilitate exchange of DNA, which is pretty amazing. So they're extremely diverse, both in form, so morphological, and as well as their different functions. Some can be autotrophic, chemotrophic, can do a lot of different things. And like in the last video, their diversity is due to several things. So they got a short generation time, so they can quickly recombine genes and have mutations. And they're also a super old lineages, so they've had a really long time to diversify. Okay, so when you put those two together, you get a really diverse group here. Some common questions we'll kind of have, and this relates to your handout and worksheet, and hopefully have a better sense for the answers to this. So this is a good time to pull your worksheet out or highlight in your handout and follow along. So questions of how can one type of bacteria be differentiated from other types? How do we classify bacteria? And also, can we differentiate bacteria from archaea? Okay, we're going to talk about each of these questions. Right, and the first is how we can classify them. So we've got this huge amount of diversity, and when we look under microscope, they're really tiny, so we don't see much. So we can use a few different ways to classify them. The first is gram stain. Are they gram positive or gram negative? Okay, if you don't know what that means, that's fine. We're going to talk about that. Their cell shape. So we can see some cell shapes when we stain them, and also their lifestyles. So, what is their, quote, relationship status? Are they pathogenic? Do they cause disease? Do they not? Are they mutualistic? Okay. Metabolic profile is also important, which we'll touch on, as well as, kind of what they're growing on, or their colony growth morphology. So here's our basic plasma membrane of bacteria. We've got our phospholipid bilayer. Okay. And like I said, the important phrase here is the fatty acid tails of phospholipids are linked by a glycerol, okay, head group by an ester linkage. So we've got our fatty acid linked by ester linkages to our glycerol, okay? You don't really need to be aware of the internal, chemistry here, the biochemistry ramifications of it. Just kind of commit that to memory because we're going to see something different in archaea. We think about the cell walls of bacteria, okay? Peptidoglycan is what we're going to be focused on, and we're going to be focused on the amount of peptidoglycan, where they have thin or thick layers. Okay, this is also demonstrating that outer membrane, which we some will have. Okay, all right. So here's the important stuff. So we've talked about gram positive, gram negative, but now we're going to talk about what exactly that means. And simply put, gram positive cells are going to have thick layers of peptidoglycan. Okay, you can see that illustrated here. And our gram negative cells are going to have very thin layers demonstrated here. All right. It may seem pretty basic to be gram positive or gram negative, but it's actually really important and very fundamental for different lineages of bacteria. So just an easy way we can group them. Okay. You'll also see here we've talked about them staining purple and staining pink. Okay. And what we mean by staining is literally colorizing them essentially. So if we've got a mixed bag of gram-positive and gram-negative bacteria, all right, the process is to dye the entire thing purple using crystal violet, and we use iodine to kind of, group that together to make it stick, all right? Then we come in with alcohol and we wash it away, okay? And what's going to happen to those thick layers of peptidoglycan is that we can't wash it all away and they'll remain purple, but those thin layers here will wash away and they'll be clear, all right? So at this point, we can't see our gram-negative bacteria. However, when we come in with safranin, which is kind of a very powerful pink color, we'll actually see the gram-negative being pink, and our gram-positive will actually stay purple because that purple is just going to overpower that safranin, okay? And what that looks like, oops, sorry about that. And what that'll look like under a slide, okay, we can actually see the little pink here is going to be our gram-negative, okay? Sometimes it can be a pretty powerful pink, like almost a magenta, okay? And also our gram-positive is going to be this purple color here, and sometimes it's even on the bluer side. A good thing to do is to, kind of watch this video for how it's done. Usually we would do this in lab. Another good thing is just to simply type in gram-negative or gram-positive bacteria on Google and start getting a feel for what those colors look like, okay? That's something that could very well easily pop up on the lab practical, and a lot of times does. All right, another way we can classify them is with the cell morphology, okay? Basically, what shape is the cell, all right? So we've got some common names, and then we've got some more, scientific names, okay? And both of these are going to be important, all right? You'll notice here that the genus of bacteria, when you start looking through those lineages, can often indicate the shape, okay? So you may see something Bacillus, yada, 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 okay? So the first shape we have is the circle, which is called coccus, okay? We have rod-shaped, which is Bacillus. We can have either single or double Bacillus. We also have crescent, or kind of, curve shape, which is Vibrio. And then the last two, Spiralium and Spirochete, we're not going to ask you to know the difference in. You can just defer to calling them Spiralium, okay? So simple things like shape. We really can't see a lot of distinctive features about bacteria, but we can see shape when we stain them, okay? And an example of this is, this slide right here, we're going to have gram-negative and gram-positive. Right. We also can see that these gram-positive in this purple are coccus shaped, so they're circular. And then if you look really close, and it's kind of hard to see in this example, but you can see that these gram-negative bacteria are actually kind of squished ovals and do have a rod shape. So those would be Bacillus. Okay. Like I said, look very close at that so you don't get tripped up. All right. Probably when you think of bacteria, you think of pathogens, you think of diseases, you think of antibacterial medications, all right? And a lot of what we know about bacteria kind of came from studying diseases, okay? So if it's a pathogenicity bacteria, what that means is it can cause disease, all right? Typically we associate this with bad things when they cause us to get sick, but in nature, it's actually always not a bad thing because it can regulate ecosystems and keeping populations under control. Okay, like we've talked about, bacteria also have mutualisms. We owe a lot to our existence to bacteria because they're in our guts right now, doing all sorts of great things for us, allowing us to obtain nutrients and live. And it's not just in humans, it's in a lot of different animals and mammals as well. The other lifestyle we'll talk about is saprobic, meaning they just break down dead things. There's tons of bacteria in soil, and a lot of times this is important for carbon and nitrogen cycling. All right, so they can release the carbon that's trapped in dead, rotting materials back into the atmosphere for plants to use for photosynthesis. And then we look at nitrogen, there's kind of two different ways they can both fix it and also release it as well. All right, some of the last ways we can classify them are metabolic tests. So, can they ferment lactase? Are they anaerobic or aerobic bacteria? Some of these other things we're not really going to talk too much about. It's just important for you to kind of be aware. They could, use citric acid as a carbon source. Can they not? So capable of a lot of different things. All right. And lastly, also their grow morphology. So what are they capable of growing on? So we can see here, we've got some different patterns depending on which bacteria is being cultured here. So we've got one that can grow on chocolate and or agar. Okay, so that's kind of interesting. Right, and why did I just cover all of that? Well, when you're going through your key lineages of bacteria and you need to be able to recall for the lab practical what group is which, okay, it's important to know which are gram-negative, which are gram-positive, what is their shape, okay? We may ask a question, what lineage is this gram-negative rod-shaped bacteria, okay? And you look through there, and we may throw something in like it's also photosynthetic, okay? And you need to use those as kind of different little filters to help key in to what group we're talking about, okay? So keep these characteristics in mind. Are they gram-negative or gram-positive? What's their cell shape, excuse me. What's something interesting about them? A lot of them have diagnostic features, okay? And that's where the red boxes is gonna come in handy when you're looking through those lineages of bacteria. All right, so that's it for activity 2. Tune back in for activity 3. And as always, email me or your instructors or stop by office hours if you have any questions.